Good evening, everyone. Welcome to Strait Science. Strait Science is a public science seminar series put on by University of Alaska Fairbanks, Northwest Campus here in Nome, and also University of Alaska Fairbanks Alaska Sea Grant, also here in Nome. And tonight, it's kind of amazing. It's May 30th, and the ice just went out. The temperature rose up to 63 degrees. It's still warm. I don't know what today's temperature is, but yesterday it rose up to 63 degrees. We switched from winter to summer in a very, very short, outstanding period of time. And the ice let go yesterday, and Nome is going to have its first ship come in tonight. So if they come in before midnight, it'll still be May if they come in after midnight. So Nome is going to wake up and see a ship at the dock in May and wonder what it's all about. And you're going to hear it first. So the ship is here to take our speakers and some of the other people in the room off to the north to find walruses to do a Pacific walrus population estimate. And we're going to hear tonight more about what they hope to learn, how they're going to do it, how they do the count, and when we'll hear the results, which I'm sure will take a while to get back. But tonight's speakers, you're both speaking. Yeah. So William Beatty, who you missed last week's Strait Science, and I signed off telling everyone Warren Beatty is going to be the speaker. Sorry about that. William Beatty, even better. William Beatty is a research biologist with the US Geological Survey, not only in Alaska, but also in Wisconsin. So you can explain that to everybody. And Irina Trukhanova, she'll be coming up at bat next. She is a contract wildlife biologist with the US Fish and Wildlife Service office in Anchorage. And she is the lead of, or you're both leads? You're both leads. Okay, and they're co-leads for this expedition to go get the skin samples needed to get a Pacific walrus population estimate. And there's other research that's applicable to this region as well that'll be going on while they are at sea. So with that, I'll hand it over to William Beatty, and we'll take it away. We're so glad to have you here.
Thank you. Thanks everyone for tuning in and for having some people here in person. It's really actually cool. So not a hybrid version of this. So as Gay said, my name is William Beatty, Bill Beatty, most of you know me as. So I'm going to take you through the first part of the talk this evening. And Irina Trukhanova will take you through the second part. So I will be covering Pacific walrus ecology, kind of a basic overview of the ecology, and an overview of the first generation of what we call the first generation of research cruises from 2013 and 2017. And from that, that group of cruises, we did estimate walrus abundance, and with related data, estimating population trend. And after that, we'll transition to kind of what we're doing this year and last year and even hopefully next year with developing new genetic methods and obtain a new population estimate for Pacific walruses. And then we'll wrap up with some future work. So like I said, I'll take you through the first part of the talk, and Irina will step in and take you through the second half. So the Pacific walrus is one of 3 subspecies of walruses, 2 to 3 subspecies of walruses. The Atlantic walrus ranges from Hudson Bay all the way over east, and the Laptev walrus in orange. It's generally considered an isolated subpopulation of the Pacific walrus. It's got limited information available. And the Pacific walrus is shown in pink, and that subpopulation or that subspecies is jointly managed by, of course, the United States, the Russian Federation, and Alaska natives. So, and that's what I'm going to be focusing on when I say walrus, I mean Pacific walrus. So the Pacific walrus itself ranges from the Bering Sea shelf all the way north to the Chukchi Sea shelf, the edge, and but the population is segregated in the summer. So in the Chukchi Sea, we get in the Bering Sea we have adult males that rest at coastal haulouts in the Bering Sea and forage on the Bering Sea shelf in the summer, while adult females and juveniles are foraging in the Chukchi Sea in the summer and resting on sea ice between foraging bouts to the bottom of the sea floor. So this gives you an idea of why we're up here sampling the Chukchi Sea. We're sampling adult females and juveniles, the part of the population that's most important for getting the population estimate and population dynamics. So Pacific walruses are benthic feeders, so they're bottom feeders. They dive to the bottom of the sea floor to acquire their prey. They're not really deep divers. It's thought that they can dive up to 200 meters, but in the Chukchi Sea, there's the depths up there are much less than that, 40 to 80 meters in the Chukchi. So they're not really that deep. They have neat little adaptations. They jet water to remove sediment, blow sediment at the bottom of the sea to be able to pull out those bivalves that they like so much, and birds as well. So sea ice is an important habitat for walruses. It's used in courtship and breeding, birthing, and nursing, and it's used as a resting platform, both in winter and throughout the year, but also in summer in the Chukchi Sea. It's used as a resting platform that walruses haul out on between foraging bouts. Their foraging bouts last anywhere, an average of 9 hours. So they are about relatively long bouts of foraging. So they do compensate for that with long bouts of resting on sea ice, which gives us access to them when they're resting on sea ice, which we'll talk about a little bit later. I do want to note that they do rest on land when there's no sea ice. So everybody here is familiar with the haul out at Point Lay, which occurs in late summer and early fall, where we have tens of thousands, even hundreds of thousands of animals gathering on shore and using that haul out to rest after the ice has melted off the Chukchi Sea shelf in the late summer and early fall. So the best information we have right now is that the population is stable as of 2015. That was the last time data was collected for that analysis, which Rebecca Taylor led at USGS several years ago. But that was, again, 2015. So we're rapidly coming up to 10 years since the last estimate of population trend was derived for this, or was relevant for the species or the subspecies. So Pacific walruses are facing several emerging threats as the environment changes up here. So sea ice loss is obviously the #1 threat. The most physically dominant driver in these systems is sea ice loss. It's creating a lot of different changes, and walruses are not immune to them at all. So sea ice loss for walruses is causing increased use of these coastal haulouts. So while walruses kind of have always been known through history to rest on land occasionally, especially the adult males, what's changing now with reduced sea ice and a longer ice-free period in the Chukchi Sea is they're just resting on land for a lot longer period of time. That makes them more vulnerable to disturbance and even disease spreading through the population quickly. So, the longer you're in a spot and in these big groups, the more likely it is you're going to get 1 or 2 disturbances that could wipe out a good portion of calves in the population, for example, or 1-year-olds. So sea ice loss is also generating decreased access to preferred foraging areas. So walruses like to forage in the northeast Chukchi Sea in a place called Hannah Shoal. It's a really shallow area in the Chukchi Sea. There's lots of local primary production there that generates a really productive benthic ecosystem. It's a good ways from Point Lay. So walruses either have to, in the late summer and early fall, they either rest at Point Lay and then swim 100 to 150 kilometers to Hannah Shoal to feed, or they feed in less productive areas closer to the haul out. So it's decreasing these access to preferred foraging areas. So they have to swim longer and maybe use more energy, or not even make that trip at all if they have calves with them, for example. So this is generating an alternate energy budget, and there's been quite a bit of work on this from some of the legacy tagging data from USGS that shows that when ice is in an area, walruses feed more and haul out more. When ice is not available in the immediate area of the walrus, they travel more in the water without feeding, and they rest less. So they're just completely altered energy dynamic there between when ice is around and when ice isn't around. When there's no ice, they're spending a lot more energy traveling, getting to places to feed, and spending less time hauled out. When there is ice, they can spend less time traveling because the ice is right there, their preferred foraging area. So sea ice loss is right now the biggest threat to Pacific walruses, but there's other things that are happening too, right? So ocean acidification could alter bivalve production or benthic production. Shipping, not so much in the US side, but on the Russian side of the Chukchi Sea, shipping is increasing. And these ships go pretty close to these coastal haulouts and during the ice-free season. This could create disturbance that could create a mass mortality event. Oil and gas development, again, not really occurring on the US side, but on the Russian side it's occurring. And of course, legacy and emerging pathogens and contaminants as well are concerned for this subspecies. So Pacific walruses are an important cultural and subsistence resource for Alaska Natives in the region. Their ivory carving is an important cultural tradition that can act as a source of income for Alaska Natives in remote villages, but also acts as outreach for the way of life throughout the lower 48. But walruses itself, is a viable subsistence resource in and of itself for communities. So that brings us to the first generation of research cruises, which occurred from 2013 to 2017. And I just wanted to recognize all the names that really participated or really were integral in developing the products that came out of those cruises. And several of those people are in the room or on the call. So a lot of work went into this cruise, and we'll talk about that a little bit when we transition to what we're doing right now. So this was a large effort from several agencies, lots of collaborators and lots of funding sources. So this project was sort of conceived way back in 2010 and 2011, when shortly after the last walrus aerial survey was conducted in 2006. So everybody recognized back at the time that there were some major issues with surveying walruses from the air, from an airplane. They gather in these really, really large groups, even when they're on sea ice, or really large aggregations when they're on sea ice, right? If you find one walrus group, you're going to find more walrus groups when you're out at sea. So that makes it really difficult to survey the population and get an accurate population estimate from aerial survey. So people back in 2010, 2011 went back to the drawing board and thought, how else could we estimate Pacific walrus abundance and population dynamics? And the goal was to set out, when this project started way back in 2010, 2011, was to estimate Pacific walrus abundance with improved precision compared to the aerial survey. Now, the aerial survey that occurred in 2006, the estimate for that was about 130,000 animals, but the analysis said, well, there could be anywhere from 55,000 to 500,000 animals out there. And it's not really very useful when you're thinking about how to issue regulations or even think about population dynamics as what's happening with the population. So we wanted to take that uncertainty, which is shown in the gray shaded polygon there, and squeeze that uncertainty. And we also call that improving precision. So one way to measure that is using something called the coefficient of variation. So higher values for coefficients of variation are less precise, more uncertain. Lower values are more precise and more certain. So we want lower values for the coefficient of variation for this Pacific walrus research cruises first generation. The coefficient of variation for that estimate in 2006 was 0.46. So our goal was to get that to less than 0.2. So we wanted to basically cut that in half and then some to see if we can get a better estimate of abundance with better, tighter confidence intervals around that estimate. So we turned to a technique called genetic marker capture. This was a technique that has a lot of advantages. You don't have to capture or tag an animal physically, so it's perfect for walruses. We know capturing them and handling them is really difficult and challenging. All you need is some type of biological sample for genetics. And in our case, we were taking skin samples. And the way this works is you take a bunch of samples from a population and you bring them all back to the lab, genetics on them. And then you compare every sample you have to every other sample and say, are they the same, basically? And if they're the same, they're called a match. It's the same walrus. And that's called the recapture and the mark recapture framework. And those are the currency of any genetic mark, any mark recapture analysis. It's kind of like your sample. You want as many recaptures as you can to get a more precise estimate. So generally, in larger population, recaptures are rare, or more rare, I should say rare, period. In smaller population, recaptures are more frequent. That's just a general rule of thumb about how to think about this. But you need those recaptures to be able to estimate population abundance and other metrics. So we conducted 5 research cruises from 2013 to 2017. The first 2 years of the project, we were restricted to US waters. 2015, we accessed Russian waters, which you can see on the map there. 2016, we were back into just US waters. In 2017, we cut back into Russian waters. So we tried pretty hard to get into Russia, especially in 2016. It just didn't happen for myriad of reasons. But so we did access Russian waters in 2 out of the 5 years of this research cruise. And the first 3 years were done on the Norseman 2, which will be the ship here, as Gay alluded to, that coming into the harbor tonight. The last 3 years were on a Russian flag ship called the Professor Molnowski. So we're doing okay on time. So
Don't worry about time.
Yeah, we'll play the video. This is just a video of how we actually conduct the biopsy sampling. So Jason Everett, who's in the room here, is on the front left of the skiff. And Casey Clark is on the right. He was a UAF student at the time.
So can you kind of talk to us about what we're seeing?
Right now, they're approaching at idle speed, very slow. The wind is blowing, so it looks like they're moving a little bit faster. And before, you kind of saw Jason and Casey were talking about which animal they're going to shoot and sample. So you don't want to sample the same animal. So it just takes a pretty brief conversation. You deploy your arrows, and the driver is actually behind him. You can see that the dart coming in the driver's shot that one. So after they go in the water, kind of wait for them to leave the area, and they'll go pick up the arrows. There's Jason. So another thing that we did on the first generation of research cruises each year was conduct age structure surveys. So, so the biopsy was one side of those cruises, and the age structure surveys were kind of the other side of the cruises. And for age structure surveys, it's a different approach. We're approaching groups of animals hauled out on sea ice, and every animal in the group we need to assign an age and the sex for that animal for the group to count in the sampling later on. So we approach animals with binoculars, a team of 4 or 3 people that is on the flybridge of the ship. So they're up pretty high. We get a pretty good view. We talk through the animals and make sure we're all in agreement for all of them. And we use this method that Bud Fay developed back in the 80s, where we look at the length of the tusk and compare it to the width of the snout to get an approximate age on the animal. So these are the sampling locations from the first generation of research cruises from 2013 to 2017 again. So 5 research cruises. And gray show the sampling locations. You know, we see most of them again are concentrated in the US side of the Chukchi Sea, because we were only accessing the Russian waters in 2 out of the 5 years. We ended up having 8,165 unique individuals. We had over 9,300 biopsy samples though. So we had a lot of animals that we recaptured in the same research cruise. So because a lot of times you can't really tell if you resample an animal when you're out at sea. You can't even tell if you sampled it maybe hours ago or a day ago. There's no visible scarring or anything to make note of. So a lot of those tissue samples, they were the same animal within that same research cruise within that same year. But we did have enough recaptures among years, so to estimate population abundance. So we finally got our new abundance estimate, and this abundance estimate applies to around 2015, so the middle of these research cruises. It wasn't actually published until 2022, and I'll talk about that a little bit later. So remember, the Pacific walrus population is sexually segregated, separate in the summer, and so we're doing all the sampling, or most of the sampling, to go back in the Chukchi Sea. There are a handful of samples in the Bering, but overwhelmingly the samples are collected in the Chukchi Sea. So we're dealing with the adult females and juveniles. And so the first thing we had to do was estimate adult female and juvenile abundance. We estimated that about 188,000 animals, but that our confidence in that was anywhere from 119,000 to about 287,000. And this is just for the adult females, juveniles, and calves, I should note as well, in the population. So we had to take that and make a few assumptions and generate a total abundance estimate, which included adult males, which we came up with as about 257,000 animals. But our confidence interval was anywhere from 171,000 animals to about 366,000 animals. So did we achieve our goal, right? Our goal when we set out was to increase precision, reduce that uncertainty, squash that gray polygon, right, in the abundance estimate. And did we hit our target for that coefficient of variation? Remember, we're trying to get it down to 0.2, a number that's reasonable in a lot of people's eyes in terms of abundance estimates for this type of species. So the answer was yes, we did. We cut it in more than half, and we hit 0.19. So that you can see that gray the gray box is quite a bit smaller than the gray box for the aerial survey in 2006, which I showed you earlier in the presentation. So we're really happy with this result. Even I was suspicious that we would hit this goal even just 3 years ago. So we're really happy when we finished the analysis that we hit the goal. So, but I did mention that this first generation of research cruises, it took over 10 years actually, from discussions and planning and preliminary sampling, developing the genetics. It took me a while to analyze the data. We had to wait for the lab to get us back. We had to do some other analyses on the genetic data before we could say, yeah, we can actually estimate abundance with genetic data. So a lot went into it to get an abundance estimate, essentially. So our goal, so that required a lot of investment in terms of funds, but also time, like time for the biologists, right? If they're working on this, they can't work on other things, basically. And so that's something that I like to remind everyone that, like, yeah, our time isn't free, right? So when we're working on something for 10 years, we don't have a lot of time to respond to other concerns. So we wanted to reduce the required investment to obtain an abundance estimate. So for this next generation of research cruises, this 10-year project is not sustainable, right? Even for government, that's way too long to get an abundance estimate. We wanted to reduce the amount of time we had to put into it. And so you're going to make it cheaper in several ways, just by upfront funds, but also cheaper in terms of the cost of the time it takes for the biologist to finish the project. So we turned to a technique called close kin mark recapture. So this is an emerging technique. It's still relatively new in the literature, but the basic premise is that it starts with, well, everybody, including walruses, has two parents, a mom and a dad. That's the very basic premise. So this method can't be used on really complicated breeding systems with other species that aren't mammals, but and birds. But that's the basic premise, and it uses the frequency of kinship relationships in a population to estimate abundance, survival, and reproduction. And so when I say kinship relationships, I mean related animals. So the closest relationship you can get is yourself, right? So that's what we did before with genetic marker recapture. So you can compare two samples and you might be the same animal. But the other relationships are parent offspring. So you might grab two samples from the population, and there might be a parent and it might be an offspring. Or you grab two samples from the population and they might be full siblings, which would be a surprise in the walrus population, but it could happen. Or more likely, you grab two samples and they're half siblings. They might have the same father, they might have the same mother, but they're half siblings. And for our purposes, we stop there. After that, you're basically unrelated. And for the close kin analysis, you could go to all the different other types of relationships that requires more and more genetic data and more uncertainty. So the catch with close kin marker recaptures, we need more genetic data from what we did before. So we need that genetic data, which is kind of the currency of close kin marker recapture. You need a bunch of genetic markers to identify these relationships. So we need enough to identify half siblings, which requires about 2,000 SNPs, single nucleotide polymorphisms, if you know the genetics lingo. So that's a lot of data for each animal. You've got 2,000 data points basically that you then use to identify these relationships. We also need to collect different genetic data to determine if half siblings are maternal or paternal. So do they have the same mom or do they have the same dad? And that's going to be relevant for population dynamics. And finally, we can use the structure of the DNA, the methylation structure essentially, but the structure of the DNA to estimate age with uncertainty. So we don't have to necessarily rely on using the tusk approach. Once we bring the samples back to the lab, we can get a point estimate for age and we can put some bounds around that. So that's really going to be advantageous for the close kin marker recapture approach. It's going to let us identify those older animals that with the tusk approach, you can't go older than 15. You just lose that resolution with that approach. So it's called epigenetic aging, but we basically use DNA structure to estimate age. So why close kin marker recapture? Why CKMR? We shorten it to that. We say it all the time. And a few more things that make it really cool is that we can use samples from animals that are alive in the Truck GC, but we can also use harvested samples. You can include dead animals in the analysis too, which we hope to do. And secondly, it's likely going to improve the precision. So squash that uncertainty around all of our demographic estimates. So not just abundance, but survival, reproductive rate, and even population trend. I think it's going to squash that uncertainty around that. But most importantly, it's going to reduce the investment required to estimate abundance and all these other parameters. So we can do this with fewer research cruises and fewer samples overall. So already do have a nice bank of samples to work with. So that's why we settled on close kin marker recapture. It does require some upfront investment in developing the genetic data, but that's a lot cheaper than doing another cruise or two more cruises. So that's why we transitioned to close kin marker recapture. With that, I'll turn it over to Irina.
Thank you.
Are we holding our questions till the end? How would you guys?
I think so.
Okay.
Yeah, we've got
All right.
a lot of related stuff.
Terrific.
Okay. Well, moving on to the second generation of cruises. So as we all know, for the Pacific walrus, and up-to-date information on population dynamics is required under the Marine Mammal Protection Act to meet management and conservation needs. And specifically, we're interested to see what the population trend is. So in order to estimate the trend, we need against two independent abundance estimates several years apart. And also we're interested to see how the population is doing under all these new stressors that Bill talked about, and what has been going on with the population since the last assessment in 2017. So in order to do a new assessment, almost 10 years after the previous one, US Geological Survey and US Fish and Wildlife Service launched a second generation of walrus research cruises. And this second generation of cruises is obviously built upon all the experience that we obtained during the first generation of cruises, and also it makes use of all of these statistical and genetic advantages that Bill just talked about. So the first of the second generation of cruises happened last summer in June. The vessel, oh, sorry, I missed a couple things. So our goal for the second generation was to, and is to conduct from 3 to 5 research cruises, and we started in 2023 last year. And the overall objectives stayed the same. We want to collect genetic information and also age structure data from the walruses in the Northern Bering and Chukchi Sea. And in parallel to that field effort, we're also working on developing a model to estimate abundance, trend, and demographic parameters. So last year, our research vessel, Norseman 2, departed from Nome, and we spent 27 days working in the Northern Bering and Chukchi Sea. And on this map, you can see our vessel route. And the numbers along the vessel track show the specific dates when we were at specific locations. And as you can see, searching for walruses, we went as far north as to Wainwright, and we spent most of our time in that area, working in the marginal ice zone over the continental shelf. We didn't search for walruses blindly. We had aerial support. So we had an aircraft conducting a search for walruses every several days, and they reported the coordinates to us wherever they found large aggregations of walruses. And then the Norseman would transit to that area, and then we will sample those animals. So prior the cruise and during the cruise, we tried to stay in contact with coastal communities. We notified them via email and over the phone calls to let them know that we were working in the area and provide communication channels so that the community members could reach back out to us and let us know if there were any concerns that our activities could overlap with the subsistence harvest. Under this project, we are coordinating our efforts with the Eskimo Walrus Commission, and due to this collaboration last year, we were lucky enough to have 3 Alaskan native hunters on board the cruise with us. We had Clarence Irigu from Gamble, Edwin Nunwu from Savunga, and Byers Gallagher from Nome. All of them were full research participants, and they provided us with great guidance on walrus behavior and ice conditions, based on years of their behavior, of their experience working with the walruses on ice. And they helped us maintain our operation safe, both for the scientists and for the animals. So the first cruise of the second generation was so far the most successful research cruise of all the research cruises conducted in terms of the number of samples that we were able to collect. Our dedicated biopsy crew collected over 2,100 biopsy samples from wild walruses, and about 1/3 of those samples came from juvenile animals, and about 2/3 of the samples came from adult animals, 6 years and older. Those samples were processed after the cruise, and currently they're at the genetic laboratory being analyzed. The age structure crew was also very successful. They scored or collected sex and age composition data from almost 800 walrus groups, comprised of over 3,500 individual animals. And all of those data will go into the population model further down the road. Apart from genetic samples and age structure data, we also collected a number of other samples for other studies. We collaborated with the EcoHubs project. This is a group that investigates harmful algal bloom toxins in the marine environment and also in the marine fauna, including walruses. So in collaboration with them, we collected walrus species, water samples, sediments, and benthos to look at the presence of these harmful toxins in those samples, and also look at the algae concentrations in the water. And I'll talk about the results of that analysis in a minute. We collected walrus blubber samples. Those come from the skin biopsy that we collect. And those will be analyzed for trace elements, toxic heavy metals, and also organic pollutants. And we did also collect walrus feces to look at the walrus diet composition, so to see what the walruses have been feeding on in the Chukchi Sea. So this slide has been provided to us by this EcoHub project group. They analyzed the samples that we collected last year during the cruise. And their main goal was to see whether there is any traces of harmful algal toxins in a walrus environment, and also what the concentration of this harmful algae was in the water samples. So based on the analysis, they found that 88% of walrus fecal samples contained this neurotoxin called saxitoxin that's produced by this harmful algae. And then 43% of the samples contained domoic acid, that's another neurotoxin. So in all but one samples, the concentration of these toxins was found to be low, so below the food safety limit. And then in clams, they found that saxitoxin was present in 93% of samples, and domoic acid was in 25% of samples. And again, in all but one sample, the concentration was low. They also looked at the densities of Alexandrium catenella cells. Those are these harmful algae that are responsible for the blooms. And they found that the cell densities in June in the Chukchi Sea were also low. So this work is not complete yet, and we don't really know what the health impact of these toxins are for walruses and what the lethal doses are. So there's still quite a bit of work to be done to answer those questions. But based on the preliminary conclusions that this group made, the walruses are exposed to saxitoxin through consumption of toxic clams. Domoic acid doesn't seem to be a concern at the moment, but of course those couple of samples that were high on saxitoxin levels are concerning. But the analysis and the work is continuing. So moving on, this year, actually tomorrow, we will be leaving from here, from Nome, with Norseman 2, and we will spend 4 weeks working in the Northern Bering and Chukchi Sea. We will have one Alaskan native hunter with us. He's in the room here, Dana, and we are very lucky to have him on board with us. This project is a product of collaboration of multiple agencies. The cruise is funded by US Fish and Wildlife Service and USGS, and World Wildlife Fund also provides the funding to support hunters' participation in the project. We are continuing to coordinate our efforts with the Eskimo Walrus Commission, and also we work with a variety of different institutions and organizations who provide logistical support for our work. And also the cruise provides a platform for many other institutions and agencies to conduct their own studies and to collect samples and data that will help us to answer a lot of research questions that have to do with the Arctic ecosystem and the changes in the Arctic environment. Therefore, we are trying to maximize the scientific value of each individual cruise. So obviously, the goal of the cruise remains the same. We're going to collect skin biopsies and age structure data, but on top of that, we have pretty ambitious sampling plans. We are going to collect sediments, clams, worms, seawater, walrus feces, to continue analysis of harmful algal bloom toxins concentrations in the environment. We'll also collect walrus feces to look at walrus hormones, to continue the diet study, and also we'll collect blubber samples, more blubber samples, to analyze them for various contaminants. On board, we'll have acoustic Doppler current profiler running that will help us to collect information on the currents in the Chukchi Sea. This year, we are planning a pilot study to look at Pacific walrus foraging behavior in the Chukchi. So far, there is no direct observations of Pacific walrus feeding behavior available, and all the observations that exist to date come from a couple of studies made on adult male walruses. So we do believe that the foraging strategies could vary by age and sex and walrus subspecies. So we are going to use a camera array system to record, to collect acoustic recordings and also video recordings of walruses foraging at the bottom of the sea. Feeding strategies have implications to energy budgets, and we hope that this pilot study will help us understand the strategies a little bit better and also help us develop the methods to collect information of acoustic indicators of walrus foraging behavior paired with visual camera-based observations. Another research question that we are hoping to address during this cruise is whether the walruses have been exposed to highly pathogenic avian influenza or bird flu. So avian influenza is a concern for Alaska region, and here it has been identified in over 30 species of birds and mammals across region. And recently we received reports that a dead Atlantic walrus found in Svalbard in Norway tested positive for avian influenza. We also know that this virus has caused mass mortalities in marine mammal population across the globe, particularly in the South America in fur seal populations, and also in North America in some fossil seal populations. For now, this virus has not been detected in Pacific walruses, but we recognize the need to monitor the population exposure to this virus. Therefore, during this cruise, we decided to collect swab samples from dead walruses if we find them on ice. And we'll also collect fecal swabs from walrus scat on ice. And those samples will be transferred to Alaska Department of Fish and Game for screening for viral diseases. And just for your information, if you see on the screen, there's a couple of phone numbers that you may use if you find a sick walrus, seawater, or polar bear. So these are the numbers that you may call. So what comes next after the cruise? We are hoping to carry out at least one more cruise in 2025 or 2026. At the same time, we have a lot of work ahead of us to analyze genetic data from all the cruises and identify individuals and kin pairs. Once that's done, I will be working on building an integrated population model to estimate abundance, demographic parameters, survival of walruses. And also, apart from collecting samples from walrus cruises, from wild walruses, we are working on an analytical framework that would allow us to include samples from high-risk walruses into our data set. And if we are able to do that, that will help us to reduce the number of samples that we need to collect from wild walruses in the future. And with that, I would like to acknowledge all the researchers and Alaska Native hunters, as well as the crew of the research vessel Norseman 2 and Professor Multanovsky for making this project happening. And we're also thankful to all the funding agencies that have been supporting this project from the very beginning. And I guess now we can take questions.
All right.
So I'm gonna
Kind of do the questions here, otherwise I'd stand up there, but I think it might work this way. So thank you both for both our speakers for giving us such a beautiful presentation as well, verbally as well as visually. So this is the time, before we get into questions, this is the time for the Straight Science audience, although the people in the room aren't going to be able to do it, but we show a little love for our Straight Science speakers. So put some love in the chat for Irina and William, and or you can call him Warren like I do, but he seems to be okay with it. And it's never easy to be a public speaker, and they just came literally off the jet and hopped in the room. We were still trying to set up. So for that, give a little love, and then we'll get right to questions. I've got some. Does anyone have a question in the room? Okay, these are all pros in here. Go ahead. I see your hand is raised, Jill. Just unmute and ask away.
So I caught the part where they did the studies on the saxitoxin in the walrus.
Looks like Jill Campbell, we can see your hand is raised. You are unmuted. Go ahead.
Can you hear me?
Ooh, oh, you know what? It probably is the volume. Hang on one sec. That's a technical difficulty on our, oh, you guys can hear. I'm gonna step out, so I'll yell the question to you guys. Go ahead, Jill.
Can you hear me?
I can, yes.
Okay. I saw the study in the walrus for the saxitoxin. What part of the walrus did you do the study on? Did you do the outer, the blubber and the meat and the heart?
Did you get the question?
Yeah, I got it. Yeah, so this during this study we just used walrus scat, so just feces from mice. We didn't analyze any tissues for the toxins.
It's gonna be awkward, but it'll work. But let me answer this for Jill. So Jill, for the question, wait, I better do it like this. So Jill, for the question of the meat and the blubber, organs and feces, that is part of the larger EcoHab study. Right now, this ship's primary focus is to go out and do the skin sampling with the bow and arrow off the walrus. They need those skin samples to do their mathematics to get a count of walrus. They are also going to be collecting unuk from the ice, and if they come across a dead walrus, I'm sure they're gonna cut into it and get some feces and maybe sample a whole suite of tissues. But for the study that's looking at does saxitoxin affect all the organs and parts of a walrus that could be consumed by people equally, that needs really sort of fresh samples, typically from harvested animals. And this year, my understanding is, and correct me if I'm wrong, I have a lot of people in the room that can correct me if I'm wrong, that there is no walrus harvest sampling program this year. So there's gonna be no regular harvested walruses sampled. However, Chuck Manade at Coeric's subsistence program, myself here at Sea Grant, and many others will be keeping an eye out for fresh dead stranded walrus, and we will be sampling them, taking a suite of tissues to give to that EcoHab effort. And as well as we will accept any harvested harvested tissues. If, for example, Savunga wants to participate or Gamble wants to participate, you can what we're really looking for is, about 3 feet of uncleaned intestine that'll still have walrus unuk or feces in it. And if you want to contribute a little chunk of meat about half the size of a Snicker bar, a little chunk of liver, a little chunk of kidney, you can put it all in a plastic bag. Like at Savunga, they're ready to do this at the IRA office. They have a freezer, you just freeze it and they'll freight collect it in. They'll call us first here in Nome and freight collect it in. So I hope that helps answer that. There is a way to get those tissues in, even without a bigger sampling program. We all wish the harvest monitoring sampling program, probably have not said that correctly, but the walrus harvest monitoring program, we wish that was in place, but we can pivot like Bering Strait is always good at doing, and we can make things work regardless. Does that help?
Yes, thank you.
Yeah. And then I gotta step out so we don't get too big an echo. Go ahead, Edwin.
Yes, hi. Hi there, William and Irina. It's good to see you guys with the presentation. And I have a question on the saxitoxin from the previous webinar that we did with the NOAA. They're gay. I remember we located, you guys described a location in certain parts of the Bering Straits where we had high counts of saxitoxin and other things possibly. And if you could explain that in difference with this 83% of feces collection samples that detect saxitoxin, is there any way that we could see the difference from our samples and NOAA samples and to have a better understanding of this saxitoxin or Alexandria algae bloom that affects the clams and stuff?
Okay, thanks. Thanks, Edwin. So, Irina, do you guys wanna, I can take a handle at this, it's another
That's an
saxitoxin
excellent
question.
question. Probably that's a question to the EcoHubs project, but if Gay, you can
Okay,
chime in. Yeah,
So,
please do.
so Edwin, that's a good question, and I'm glad there's so much interest in saxitoxin. It is a real concern for us. So in my understanding, from the slide that Irina showed, they were sampling the Norseman 2 crews were sampling up in the northeast Chukchi Sea. They sampled, I believe it was 42 walruses, roughly. And out of those 42 fecal samples, 88% of the walrus detected there was saxitoxin, low level, except for one sample. So out of 42 walrus, yeah, we know we have saxitoxin. We know it's around low level, but one walrus, and that was up in the northeast Chukchi, right? North of Bering Strait. Is that correct?
Yeah.
Okay.
Mhm.
So, so the samples that are collected here in the Bering Strait region will go to that same project. And we can make sure that when we have if you see the screen, the people on the top there, especially the woman on the left and the man by the sign that says Woods Hole Oceanographic Institution, they'll probably be back giving us straight science with the results of a lot of the walruses they're getting samples from, like this cruise and others, and we'll be targeting a harmful algal bloom, the results of their work. So that's where the northern Bering Sea samples and the samples that were collected in 2023 on the Norseman by you and Clarence and all the researchers, that's probably when we'll see that comparison. That is in the works. Those are all going to the same project. Does that answer your question?
Yeah, is it the water samples that was giving us the data on the counts per so much, like within the certain depth, or I forget what we were talking about on the we had high level of detection in that area, in that one area where it was real
Danger.
large number of count, yeah, in the samples. And I just wanted to find out if any of that weigh in with the feces samples that we took there on the cruise.
Yeah,
Okay, answer again.
so all the water samples that were collected were tested for this densities of algae cells, and they were all low. So there was no indication of any bloom happening at that point in the Chukchi Sea.
So
Okay,
did you
thank you.
have on board the Norseman an imaging flow cytobot? Okay, So you are right, Edwin, for what happened in 2022, right off of Gamble and Sabunga and Diomede and Wales and right on up into Kotzebue Sound. That very record-breaking, unprecedented large number of cells of Alexandrium, the harmful type of algae that typically is low in number, but now is under certain conditions with warming, we had this problem. You need two things. You need to know what the animal has eaten or is, he's exposed to. You can get that through the feces. And it's good to know real time what is going on in the water, so you can kind of estimate, oh my gosh, it's going to be sucked up by these clams. So it sounds like for the 2023 Norseman cruise, you were able to get the feces of the walrus and maybe some water samples that were later analyzed. What happened in 2023 and again in 2022 near St. Lawrence Island all the way up into the southern Chukchi Sea, as well as the 48 hours of information that we had a second, not as large bloom in late July of last year in the Strait. That was because large offshore research vessels have that automated water sucking machine that sucks the water in and spits it out, but it's running it by a camera that's taking massive amounts of photographs very quickly, and it has a robot that's identifying the algae in those pictures. It then can alert via the internet real time when the ship is encountering too much of a certain type of algae. So fortunately, we will have 2 ships this summer that will be in the area passing through the Bering Strait region, the United States Coast Guard icebreaker Healy, and later on, and also Sikuliak will be coming up and down, I believe twice. They will have on board one of those automated machines. We will get information real time on how dense the Alexandrium cells are, but we probably won't have any walrus samples at that time unless they're harvested. Does that help?
Thank you very much, Gay, for the explanation. That was that Alexander bloom that had high count, and so it does become saxitoxin once it's filtering through the clam and it's eaten from a walrus. Is that correct?
So the Alexandrium, which is the name of the type of algae that's now growing more because we have warmer water, it naturally just produces saxitoxin. Actually, it produces a mix of toxins. The worst one is saxitoxin. So if you have a lot of Alexandrium in the water, you may have a lot of saxitoxin that could affect the health of warm-blooded things like seabirds and people and walruses and bearded seals. If the prey that those animals have eaten or people have eaten has too much Alexandrium algae in it. So like a clam's filtering algae all the time, you have a big growth of Alexandrium, that clam's going to be full of Alexandrium. Walrus eats the clam, he's going to get a dose of the poison. Or toxin. Is that helpful? I don't need to go on, but.
Thank you very much, Gay. That was explained. You explained it good. Yeah.
Okay. All right. Thanks, everyone. Ask any other questions. We'll see you. All right. How about in here? Any other questions? Yes, sir.
Wondering who uses this data beyond academics. So what kind of policymakers, what kind of decisions are made based off of this, or who benefits from?
Well, I'm not the most appropriate person to answer this question, but so the cruise is jointly run by USGS, which is the agency I work for, and the Fish and Wildlife Service. So the Fish and Wildlife Service has a program in Anchorage in the regional office focused on developing regulations for marine mammals. So this abundance estimate is the most important thing for issuing those regulations.
Are those harvest regulations or interference
Oh
regulations?
no, they're called take regulations. Yeah, good question. They're called take regulations. So whenever you and there's various ways to take a marine mammal, but it could be as something as simple as like causing a behavior change. So like for the research, we actually have a permit for take of marine mammals because we are causing a behavior change. We're taking the ship and they're going in the water, right? So anybody that does any work will go through the Marine Mammals Management Office in Anchorage, any commercial vendors actually, to get that authorization, legal authorization to take marine mammals. So
would that affect shipping lane permits or oil and gas permits or?
well, oil and gas when it was active up here, but ships don't necessarily apply for those permits. Some might. It just depends on what they're doing. So Joel might have something to add to this. So he's more appropriately positioned to answer this question.
I work for the Office of Marine Mammals Management, which is in Fish and Wildlife Service. So Fish and Wildlife Service has trust responsibility for walruses, polar bears, and sea otters. And so we're the active management agency. And most of our management efforts are sort of guided by this Federal Marine Mammal Protection Act. But almost everything that we do with respect to management at some way or another is linked to abundance and demographics. So we regularly report on the status of the Pacific walrus population. That's sort of our goal, to look at numbers of animals, the size of the population, and that and trend of the population, look at emerging threats. Having abundance allows us to estimate what potential impacts are there. And then, of course, we have a lot of subsistence stakeholders that are dependent on this very important species. So, depending on the status of the population under various regulatory frameworks, it could have management implications there as well. So we're working to try to develop modeling framework to evaluate the population status. Most recently, we have a petition to consider whether or not to list walrus as an endangered species under the Endangered Species Act. And so we're evaluating available information on the size of the population, the demographics of the population, and comparing that with current threats and emerging threats to sort of figure all that out. So we're a federal agency. That data is there for everybody. It's not squirreled away in academia. It's out there. Did that help?
Well, I wasn't trying to be flippant or.
No.
Yeah,
Yeah.
Question.
I'm just
It's a very good
wondering
question.
basically who the stakeholders are, and so this data is kind of basic research and baseline studies that can be used to monitor changes over time.
Population status and trends. Yeah.
Yeah, also to add to that, for example, over in Russia, in Chukotka, the harvest, the subsistence harvest is based on quotas. So those quotas are distributed every year among different communities within so-called total allowable catch. So this total allowable catch is being estimated each year based on the best available abundance estimate available to us. So those data are used directly in this harvest regulations across the Russian waters.
There's no current harvest regulations on subsistence hunters in Alaska, federal regulations that is. So there aren't any sort of quotas or limits. There are some self-regulatory limits that the tribal governments are starting to develop, and this information is intended as a benefit to them as well, to help inform their management decisions at a tribal level.
And we have, is you guys good?
Yeah.
You're good? So we have another question from Jill Campbell. Go ahead, Jill.
Okay, so the slide after the harmful algal bloom, it talks about the avian flu being found in the walrus. I'm assuming that's, I mean, I don't know what part of the walrus was tested for the avian flu, but how is that going to be a risk for human consumption that the walrus had tested for positive for the avian flu?
One thing I will point out is that it was detected in the Atlantic walrus. So it has not been detected in Pacific walrus yet. So I will point that out. In terms of consumption, we are not qualified to answer that question. That may be something better for Gabe, maybe can chime in on, or state officials, or that's their purview.
No,
Yeah.
well, the Department of Public Health, but
Yep.
so how do you test for avian influenza? So how did they find out on the polar bear at Utqiagvik? So the North Slope Borough has their own veterinarian, they have their own wildlife department, they can pay for that up north. So the bear was healthy, fat bear, near town, no gunshot, no fight marks, like it hadn't fought. So why? This was in late October, and the vet is very good up there, and she got in all her protective gear, and she went, and you take, just like you would for when the COVID was on us, it's a virus. So you take a big long Q-tip and you shove it. Remember, if everybody remembers getting the Q-tip up the nose, that is what you do to the bear. You, or any animal you want to test for a virus, you put a Q-tip up the nose. And you also, which for a short time, if everyone remembers, you can also, if you're not wanting it up your nose, you can get it up your butt. And you can get a rectal swab, and the nose one was much more popular in the human world, but you can get a rectal swab. Both of those actually tested negative for that bear. The vet was so good, she cracked the skull of that bear and swabbed the brain. And that is a really good place to test for bird flu. So basically, you will see someone swab up the nose, just like a COVID test, up the butt, or now you want to swab the brain. Can the animal be headshot? Absolutely, it's okay. It doesn't need to be the smooth outer covering. It can actually have the brain material on it, and that's not a bad thing. Currently, there is no monitoring and surveillance, I can say that confidently, for avian influenza in the Bering Strait region, whether it will be in seabirds or marine mammals. I do believe the land animals, like a fox, for which this region has had avian influenza in red fox over at Unalakleet, and in seabirds here in Nome. There is nobody doing surveillance and monitoring. However, the Bering Strait region will, Fish and Game, the Department of Wildlife Conservation here in Region 5 in Nome, for animals that are rabid, we've had a lot of outbreaks of different things out here, but for rabid or seemingly acting distressed with seizures, that animal will be tested for avian influenza. We were about to have a man from the USDA. I don't know if you guys all saw the sign at the store for 10 days. He has canceled. So we have Chuck Monade at Coweric's Department of Subsistence and myself have swabs, and we will be, if anyone has any concerns, we will swab birds, we will swab people's catch, we will swab dead, stranded animals that are called into us on the beach. If anyone sees either birds or land animals or walrus, seals, and sea lions, the signs of avian influenza in birds, the symptoms are when the bird's spinning or walking in circles, staggering, disoriented, may have clear liquid coming out of the mouth, can't fly, approachable. what is it gonna look like for a marine mammal? Judging from marine mammals globally that have had avian influenza, they may have tremors or shakes, like a little seizure. They may have discharge coming out of their mouth and eyes, extra discharge than normal. They may be approachable, disoriented. And if you see anything like that, oh, and the marine mammals may be having difficulty breathing or coughing. The seizures usually based on marine mammals globally, and this is seals and sea lions. I don't know the situation, how the walrus was presenting itself in the Atlantic walrus that was way over on the European side of the house. But typically those seizures are happening right before the animal will perish soon after that. That's sort of the last shaky bit, and then it goes down. So if anyone sees anything unusual, we really are on our own this year throughout the region, but we're doing it ourselves, right? The Bering Strait region, we are strong and resilient, and we will come up with ways to get information. So record, use your camera as a weapon. Take photos, take videos, and call a host of people. You can call the Fish and Wildlife, you can call NOAA, you can call any of us in NOAA, Northern Sound Health Corporation as well. As far as how will you know it's safe to eat, that was part of your question, is that right?
Yes.
We have a question here too.
Yeah, okay. As far as it being safe to eat, the only guideline I can officially give you is use your best subsistence practices. If you see the animals looking not right, then go with what you've been taught. But if you don't have any idea what's going on, if you cook, it's a virus, so it's a little easier to take care of than saxitoxin. You have to cook organs or any other, even the eggs, if you're concerned about birds, marine mammals, anything that might have avian influenza affected, and you're not sure, cook it to over 165 degrees. That will blow up the cell walls of the virus, and it will be dysfunctional after 165 degrees. Hopefully that helps. Any other question on that?
I have a question.
No, thank you.
There was a question in the wrong page.
Okay, hang on one second, Edwin.
I think while she's doing that, because just figuring out
Okay,
how Atlantic
this
walrus.
question is on the avian influenza.
So they never
And,
were able to prove it getting sick. They found a dead animal and swabbed it.
One sec, Edwin.
And how, it would be interesting to know how it was transmitted, how it got it.
Yeah. And yeah, they don't know. I mean, they had seen, I think it hasn't been published yet, but my recollection of the account is that there was several carcasses in the region, which is not altogether unusual, but there was a fresh carcass. They had access to one, and they did swabs, and they sent it off. They didn't do a necropsy though, so I think my interpretation from the vet, to Dr. Raphael Stibelmeyer, is that it was likely shedding virus, and so it could very well have been the cause of death. But they without the direct observation or pathological, it's not 100%, but it was sick with avian influenza, and they weren't able to get the exact strain of it either.
Yeah, that was uncertain.
Correct.
Yeah.
So the sample was a little bit it wasn't as great as they hoped for.
Yeah.
And so because of that, they can't really say too much, is that right?
Right.
Yeah, with detail, as much detail as they would like.
Right.
I don't think they anticipated it, I mean, honestly.
They did have several bird die-offs in the area that were sampled and attributed to H5N1. So, seems like
Walrusy birds. All right, Edwin, sorry for this back and forth.
Okay, yeah, I have a question on that avian influenza. Is it the same? How many different types of avian influenza is there? Is it just one, or is it in this particular type of virus? Is it something that we should take samples on our harvests on, as far as nose swabs or brain swabs you guys talk about? And another thing is, when we give brain shots, we have opportunity to get into the brain to get a swab off that. And how can we help ourselves in the community to learn about, this avian influenza that could affect the marine mammals also?
You want to handle that?
Well, I will make.
Go ahead, this is Joel Garlic Miller talking to respond.
Hi, Edwin. It's Joel here. There's one thing about avian influenza, and it is a potential epizoonotic disease where humans could potentially get it. So people who sample suspected cases of it are having to wear quite extensive personal protective equipment and use proper and safe procedures, because you don't want to get sick. So I think the general recommendation is to call in suspected cases. Like if you see seized walruses or unusual number of dead walruses that have no bullets or anything, then the way the structure is supposed to work is we call into that event and then a response team could go out and do the sampling. I wouldn't encourage people doing that on their own.
That's for good advice. If you see an animal that you think may be sick. So if you see something that's got shakes and tremors and drooling and doesn't know what's going on, you're not going to harvest that, I don't think. You don't want to be anywhere near that one, but you want to whip out the camera for that kind of thing. Take video of what is happening to it. For animals for which you want to eat and you think they're healthy and you want to double check, I will send you swabs and we can talk about that offline if that's helpful. And that would be wonderful, Edwin. We wouldn't want you to go at animals that have got, obvious distress. We definitely would want to report photos or video of it, but leave it be. But for the ones that you're worried about consuming, I have no problem with sending you some of those Q-tips and the little vial at the top of the Q-tip once you've run it around in the brain, and you can COD it or freight collect it back. So let's talk more about that off out of this meeting.
Okay. And then another question is vaccination. Is there? I took avian bird flu vaccination before, and do they have vaccination that would be applied if it's necessary for people?
So these are really good questions. These are really good questions, Edwin. There is a vaccination that the government is working on right now. I can't think of the name of it, like Tamiflu, but I don't think that's it. But there is a vaccination they're starting to work on, so I think people are taking this, the government is recognizing this is spreading into more areas. It is global. So stand by. But yes, they are recognizing you're going to need a vaccination probably down the road, and they are working on that at this time. We do have people, there's been 3 cases in just the last few weeks at the dairy farms. There's a 3rd case that was announced today of workers, human crossover from the dairy cattle that have avian influenza. And I think these are some of the first mammal to human transmissions of avian influenza. at least in this country, if not the globe. And the people are getting healed, you know, they're healing up from it. So it is so far, given the right medical care, they are able to make a recovery. So, but this is a virus, viruses change, and these are sort of, we're just all watching this unfold, so, but thank you for your question.
Thank you.
Okay, there's another
Thank
question.
you for also sharing. Thank you for also sharing the
Go ahead.
boiling
Okay.
temperature for cooking.
It's
There's
great
the
to-
microphone.
Yeah,
It's
okay.
perfect.
It's great to hear all the concern about the health. So I'm glad
Yes.
you're doing it, okay? Thank you.
Oh, and Chuck and Norton Sound Health Corporation, it's a 4-alarm
Yeah.
thing for
It's
us.
getting the word out.
And actually, that was gonna be one of my questions, because I didn't know we were gonna get all these questions, which I'm really glad we did. And I have a question, but I think you're the sleeper, you're the boss?
I mean, no, but I won't blame.
You'll let him, okay, so here was my thing, because it came up, and my question is, I was listening in, and we understand, doing this estimate is very important, and,
We had a question.
oh, I'm sorry.
Yeah, I was just
No, I didn't.
prefacing like,
Oh, you were practicing, okay, sorry.
so I, probably where the question you're getting to, but, now we have a baseline. So the baseline for the population is the 2017.
About 260,000.
Oh, yeah, nice. So anything, what you're doing now is, will establish another baseline or another count, establish a trend.
Yeah, so what we're not doing, I'll start with that. Actually, it's probably a little bit easier to explain. So in the statistics behind all this, let's go all the way back to here. What we're not doing is connecting the dots. We're not doing that. In the close kin market recapture model, which will have other information in it, it'll eventually have age structure data, it'll have other data sets in it. We're specifically going to be estimating essentially what the population is doing. Is it going up? Is it stable? Is it going down? And then, so because we're estimating that, we can then say how confident we are in that too. So we can say, right now, you know, the best available information says we're pretty confident it's stable essentially. You know, there's some uncertainty around it, but it's basically on the margins. You know, it's not. So we won't be connecting the dots. So but we'll be estimating population trend in and of itself, like as its own thing.
You're not actually establishing a new walrus population estimate.
Oh, no, we'll have that too. Yeah, but that the walrus population estimate will be generated as well. But
okay,
yeah, but it will also at the same time be estimating if the population is increasing, stable, or decreasing.
well, obviously, if you're establishing another number, then that obviously is.
We won't be connecting the dots. So I'm saying in the model.
Well, that's what I asked you. So
Yeah.
This last one you did is not a baseline.
It's not a baseline in terms of like generating an estimate of population trend. No. So yeah, if you connect
Because the method is different?
that's what I was gonna get at. So
Okay.
like if you look at here, like, oh, the walrus population like went way up if you connect these two dots.
Well, I'm not worried about 2006.
I know. I'm using this as an example,
Oh, I see.
right?
Okay.
Right. Yeah. It's just an example but we can't do that because the methods are way different and they're more different than what we're going to be doing down the road. But that's just a good example. Like these methods are too different. You shouldn't generally connect the dots in any sort of to analyze population trend anyway. There's statistics that let you specifically estimate, just like abundance, how you can get a number, you can get an estimate on what it's doing. And actually, it's a number. It's and how you do it. If it's above 1, it's increasing. If it's 1, it's stable. If it's below 1, it's decreasing. So you get a specific number with uncertainty around it. So, so the 2015, we didn't have enough recaptures to estimate, or we didn't have a long enough study to estimate population trend and enough recaptures. So, because walruses are, you know, they're a species that they live a long time. They have relatively few offspring, so the population is not going to change a whole lot in 5 years. It's going to be pretty much the same as it was, 5 years to start. So you need a longer time period, a longer window, and close kin mark recapture, this new method we're doing, will give us that pretty long snapshot, because we're going to be going back, because we're doing pair and offspring pairs, so back 20 to 30 years in terms of the population trend. So
there are some basic differences between the approaches, though, in that closed kin, you usually get estimates of adult abundance, so breeders. So then you have to make assumptions about population structure and early life survival to sort of fill in the whole population. So and then there's also maybe some differences having to do with the spatial structure of the population too, and what you're actually estimating that might be a little bit different. So I can see why there might be some reluctance to want to connect those dots exactly, because you might be estimating a slightly different thing.
slightly different thing, yeah.
And
And so
what's your name? I'm sorry.
Oh, I'm Paul Kahn from NOAA,
Okay.
Yeah.
Oh, okay. Thank
yeah,
you.
so Paul's working on close kin with other
with bearded seals.
bearded seals, yeah. So that's a good example. So for this last estimate, we really were estimating juveniles and adult females, right? And so we had to make an assumption about, okay, if you know how many adult females there are, about how many adult males are there going to be, and you know, we took our best guess, educated guess, and so we got a total abundance estimate, because that total estimate is so important for regulatory purposes and for what marine mammals management actually needs. So, because that's what the law says, like the Marine Mammal Protection Act says total abundance. It doesn't say breeders or, you know, juveniles and adult females. It wants that total number. So all the time we're taking these methods and making some assumptions to get to that total abundance estimate too. So, good question.
Yeah, I guess I still, maybe it's all going over my head, but I still am a bit fuzzy on how establishing trends that Fish and Wildlife is going to use to make decisions about, or not making decisions, but regulatory decisions, and how it's used in the ESA assessment.
Right. Well, yeah, I mean, we are expecting that the population is going to decline here. I mean, with the shrinking habitat and through some of the processes that Bill outlined, you know, in terms of energy budgets and things like, and access to foraging beds, it seems logical to think that the carrying capacity, the total number of walruses that this ecosystem could support, will be smaller in the future. And so trends don't necessarily have to go off a cliff to zero. I mean, populations fluctuate quite a bit, and so we can model and project about what we think the population is going to do in response to changing energy budgets and things like that. But periodically, you need truth or data to sort of make sure those.
That's kind of my next question. So you're doing one more after this year, and then kind of.
Yeah, so that I was actually gonna mention that. So that's a good point. So the better way to think about kind of how we're approaching this generation of cruises with this close kin marker captures, that's going to give us sort of a baseline in terms of how do we collect more or collect fewer samples, but also be able to update that estimate through time to get us better information and frankly cheaper on abundance and population trend and other parameters. So a better way to think about it, I think, is to think about this generation as that baseline that hopefully we can build on through time. The other good news is that all the samples we collected from the not all of them, most of the samples that we collected from the last generation, we sent off to a new lab. So those will be used in this next generation of analysis as well, because it does go backward in time, more so than what we did before. So I think the baseline is what we're doing right now. It's a better way to think about it.
Great.
So, yeah.
Thank you.
And in the chat, since you can't see it, Rebecca Taylor writes, "Hi, Paul!" exclamation. Got a shout out right there, just from your voice.
That's good. Thank you.
So,
Yep.
I was just going to say that subsistence harvest is not, as I said, is not a regulated at this point. There's no sort of federal structure for regulating it, barring a total collapse of the population that under the current thing. But there are other things that are regulated, like fisheries and stuff like that. And there, so part of our management structure is to develop these stock assessment reports, and so they compare removals of from human caused removals from the population, typically fisheries, and compare that against current abundance and things like that to sort of characterize whether there are concerns about is it exceeding the the safe removal level. And so this sort of data underpins that sort of thing, and along with estimates of removals, and that can trigger some regulatory actions, greater coverage for fisheries observers and things like that, depending on the outcome of those things. So that's a practical use of the data. So yeah, all marine mammal species in the U.S. waters go through this process every 1 to 3 years, depending on whether they're strategic or non-strategic under this framework, and it would trigger management plans to address concerns if necessary.
When does the paperwork go in for the listing Joel, your office needs to answer to for whether or not walrus they have to go to a review to see if the population warrants being put on the endangered species list or not? What's the date that's all due for you?
Yeah, well, I didn't want to. I would encourage topics for our guests, but yes, at issue, the Fish and Wildlife Service is currently preparing a species status assessment, evaluating the current status and emerging threats to the Pacific walrus population. It's putting together this document that it's a science-based document. That document doesn't make a determination whether listing is warranted or not, but it will provide the science for a decision. And we have just completed our first draft of it. I mean, I think there's some polishing, editing going on by some of our colleagues. Irina's lifted heavy weights on that project as well. So that is going out through a process of peer review. And I think it's in August that that peer-reviewed information is presented to a decision team for Fish and Wildlife. They're not a decision team, they're a recommendation team. So we will have regional representatives that will review the data, query us on the data, and then they will make a recommendation to headquarters about the petition to list walrus, whether it is warranted or not warranted.
Right.
Yeah.
And what I'm doing in the chat box is Joel actually gave us Strait Science this January on this process, and there's the link to it in the chat. So that's a good way if you want to learn more. You can hear Joel Garlick Miller and Dan Bjornley of the US Fish and Wildlife Service Marine Mammals Management Office talk more about this. So I would highly recommend after tonight, if you're interested in that process, click on that link. I have a couple questions. Oh, go ahead, Edwin. Wait, I gotta get out of the room because the microphone seems to go nuts. What's your question? Go ahead, Edwin. Oh, I'm gonna unmute.
Okay, I was speaking without the mic on, but yeah. So it's a genetic question on the population status of the walrus. Besides looking into our harvests and the way we, I know it's important to look at the population, the whole population itself, and what has this administration done to question, like as far as like what things that are coming this way, like the climate change and the ice receding and the traffic vessels that are climbing the numbers, and the trawlers, all that stuff that can affect the population, and how would you be able to share the information to the community, as far as some things like the climate change and other things that can affect the climate change, and what has this administration done to question some of these things to our politics?
So the question was about what the administration has done for
all the other things, and it's kind of, I'm actually gonna ask a question after you, Edwin, because it's along the same lines, I think. I think he's asking, and tell me if I'm wrong, Edwin, you're asking, all the other stress on walruses, ships and contaminants and things like that, I don't know if Edwin means at the Biden level or at the Fish and Wildlife level.
Well,
Go ahead.
The question goes to the population, like if they're gonna put it into endangered listing, what has this administration done to question some of the things that are affecting our climate in this area and in the Arctic, as well as all the other contaminants that are flowing up this way?
Okay.
If you mean these things being also
Right.
considered.
Edwin,
That's a question, maybe?
This is Joel, just a clarifying statement that Pacific walruses are not listed under the Endangered Species Act at present. We've done one analysis that ended in 2017 and decided that listing was not warranted. That has been challenged in court, and as part of a settlement agreement, we've agreed to do a new analysis, incorporate new information. So that information's been gathered. I don't know what the outcome of that analysis is. So, but presently, the walrus population is large, healthy, harvest levels are sustainable. It's sort of an interpretation about the long-term future of it, and I don't know how the decision will go, but it's looking at the same information pretty much that we looked at the last time. It's our responsibility to communicate with you everything that goes into that analysis. We've tried to do that, sharing some of that information of how our models and projections work, either in reports or community meetings. Always need to do more about it. As far as administration, that question is so broad, I'm not sure that we can really touch on that. I mean, climate change policy, and there are, shipping, traffic, the world is just moving so fast, and there's so many parts and pieces and different regulatory bodies and agencies that Fish and Wildlife Service certainly can't sort of answer for those things. So I sense your frustration very much on all the changes you're seeing out here and things. Our small part of it is trying to manage walrus as a resource for future generations of hunters, and so that's all we can really talk about with you today.
All right. I see no other questions right now online, so maybe I'll get a chance to ask my question. I've got a couple. So I've got two for you guys right off the bat. And now, since we have guests here that are answering questions, that would be wonderful. So the first two is for the mark recapture stuff. Who's doing the microplastics at UAA with the feces? Do you know who's the
Patrick Tonko is the lead of that research group.
Patrick?
Tonko, he is with UA.
Okay,
UA.
so interesting. That's good.
That's the project. It hasn't started yet. They applied for funding. They're waiting decision.
Ah.
So we are just collecting the samples. The future project, hoping that it's going to be funded.
And then for the ADF and G, so the state of Alaska, their contribution is to the analysis of the avian influenza.
Correct.
And that'll be through the state lab that must have a contract with.
Well, that project is led by Lori Palasiak.
And that's for marine mammals, or is that the state contract for birds? Or because there's multiple avian influenza specimens coming in. Are you saying that the state of Alaska has the contract, Arctic Marine Mammal Program, or the Marine Mammal Program of the state of Alaska has the contract to do HPAI in marine mammals for Alaskans?
I'm not saying that.
Okay.
That's a smaller specific project.
Okay, but they're the all of a sudden the ones that are in charge of handling the avian influenza results.
They will be handling the samples, yes.
For this cruise or
For this
all
For this project.
Okay, that's super helpful.
They got funding.
Yeah.
That's super helpful and very interesting for the region to know. And then the other one is, I've been trying for years, I stranded responder, and now we're getting more people on board for that. I would love to give you the skin samples off lots and lots of walruses that we have responded to in the hundreds sometimes over the years. Do you want skin samples from, it sounded like you did. I was excited. Do you now want stranded or dead stranded? We respond to those regardless of who's paying the bill at the federal level. So do you want skin samples from stranded walrus? I'd be happy to give you.
Fresh.
If they're preserved and they're frozen, or
And does the walrus have to be in a pristine shape, or can it be
Fresh.
for the genetics? Okay, of the skin, but the skin only. That would usually be fresh if it's been in the water. And
yeah, it's gotta be fresh for the age. That's what it needs to be fresh for.
But-
So to get age from the structure of the DNA.
I can take a picture of the walrus and you can
Yeah,
say.
so we should talk about it.
Yeah, that would be wonderful. We've been trying to do that for years, so that may be progress. And here's the one that's for the other fish from all the. I heard you say, and I totally understand why you said it, because you're immersed, both of you, with your research backgrounds, with your genetics, and pulling off these expeditions. You are immersed in this population survey effort. That's what you do, right? But when I heard we don't have time to work on other issues, I totally understand that. But tonight, from just the callers and the questions, you can hear, and this isn't meant to you guys, and you're not supposed to answer this, so I don't think you could, but you can hear there's much interest in other issues. We have avian influenza in this region documented, right? On land animals, the red fox, in glaucous skulls, and this is with minimal sampling. We are hearing more about, its spread. Harmful levels. Thank God there is research being done on that. You guys are helping out big time in that. That is wondrous. But it's a thing that this region had the largest, I think it was most massive, most persistent, most toxic, most dense cell count nationwide ever for this Alexandrium. So that's like the us that live out here, and the walruses that are eating the clams just like we are. That's a big deal. So we're hearing this, we're seeing the ship traffic, there's a lot of other issues
I understand you are regulatorily mandated by the laws of which your bosses are telling you what to do. You have to do a population count, right? You have to do a stock assessment. You have to do a whatever it's called for, what do they call for the listing?
Species status.
Species status review or assessment or whatever it is. So when will there, do you see when might there be time to work on the other issues that are building quite rapidly? Everything from disease that affects people to problems that could affect walrus. You know, we have these military drills now, close 16 miles from Gamble. Boom, you know, they're lighting off missiles and things like that. We have large ships. When might we have time? Given all that you guys are constrained, I'm not saying you're driving the ship, but when do you, is there time soon that we may, will it be after the last walrus cruise in 25 or 26? When will we be addressing walrus other issues?
Okay, I'll say one thing that so our Fish and Wildlife Service has management responsibility for the population. So I work for US GS, which is the science agency. So some of these things have been addressed or tried to be addressed. So the shipping, for example, there was a recent paper that came out that we looked at the data we had for the effects of vessel traffic on walrus, right? And we didn't have great data. So that was mostly what we learned, but we took a step in that direction. So even already we're thinking about what the next 5 to 10 years looks like for that particular question because of the emerging concerns. So some of these questions have been addressed, or we've gotten some momentum from previous data sets, but we do need, for that one in particular, we need a new type of data, we need GPS data on walruses, which is really hard to get. Just hard logistically for the tagging. So that's one thing I will say is that so the stuff is on front burners in a lot of cases. That's an emerging project that we're having discussions about now. So go ahead, Jim. On that.
Thank you very much.
Yeah.
William.
So USGS is a research branch, and they have been very responsive to Fish and Wildlife Service needs for walruses, but they're also very interested and responsive to addressing research questions posed by the subsistence community, and they've been very solicitous of ideas for research and are doing lots of other things besides population assessment. Developing tools and techniques for monitoring the size of coastal haulouts, looking at mortality in coastal haulouts and things like that. These are all suggestions that people wanted us to prioritize. We do, and it's a reality. There's other feds here too. There's limited funding and resources, and so we have to, constantly go through a prioritizing process to focus our efforts with the time and treasure we have available to us, and we do the best we can. Our scope, it's not words you want to hear, but our scope is somewhat narrow. I mean, we are about walruses and the health of walruses, of course, and we also have mandates for co-management of subsistence use, and so we spend a lot of time and resources on that, but we aren't going to be in a position to take the lead on big oceanographic studies to address harmful algal blooms. If we can fit in things, of course we will. But the unfortunate answer is that there's many other parts and pieces out there, and I'm not sure that we are that point of the spear on some of these disease issues and things like that.
So for the audience, because they were getting a bad echo, which we were not, so that's gotten fixed. So for the audience, the question was, given that, understanding that these guys are wrapped up in population estimate, hopefully tonight they've heard, because I certainly heard, that there are many, many other issues regarding walrus health, conservation concerns, ecosystem concerns regarding walrus. And that when we heard the words, we don't have time to work on other issues, there's nothing bad with William and Irina. They're trying. Their whole job is to get this survey off the ground and get a count. But my question was, and you guys have just been tuning into, the answer was when may we have time? When will there be time to address some of the other concerns?
Well,
So,
yeah, even with these cruises, you can see that there's a lot of overlap between the concerns
Yes.
that have been voiced today and the thing that we are trying to do as a satellite projects to our bigger project.
I'll narrow that.
Providing the
Yeah.
platform and opportunities for other groups, collaborators, partners to try to answer all of those questions. And some of them, don't have funding at the moment. Some of them
Yes.
don't have resources at the moment. We are trying to collect data, trying to collect samples, and slowly in the future, hopefully soon, we'll get some of the answers that you'll
Yes, and without you guys, you are providing the only information we're getting. So we are really grateful, and everyone should be grateful to this research effort. If you live in the Bering Strait region, where you are headed is not here. And so we are grateful, because up north, they're gonna need that data on the clams. They're gonna need that data on the cyst beds and on the everything. And so for this region, which is, we're the largest consumers and users of walrus in the country, there's a lot of interest in what's happening right here in the Bering Strait region. So things like monitoring and surveillance, there are things now that are serious threats to everybody. And so my question, maybe I didn't say it right, but when do we get the biologists back or bring biologists from the region up through the ranks, that kind of thing. Just food for thought about when we can get back to looking at walruses here in the Bering Strait region. We are so grateful for what you guys are doing and that you are collecting all these other samples, because look at your track. You're going where all that offshore data is absolutely unavailable to anyone unless it was you guys doing it.
So the walruses that are up there are the
Yep.
same walruses that are here. So
We understand, but
yeah, so in terms of all the population information, that's relevant, right?
Yeah.
But the physical samples we're collecting, yes, you're right. But the for the water samples, right? But the fecal samples that could give us, we're not far. They're not far in terms of their annual cycle from this area, right? And so right now they're not far.
from where you are right now, if you're collecting them in the Bering Strait region, but I believe you guys are headed north, aren't you?
Yeah, but who knows? Last year we had, we don't have the samples on the map, but it was a lot closer to this area because of the ice conditions.
Right.
So it varies with ice conditions each
Yeah,
year,
of course.
right? And so if I showed you the map of the sample, so there's a lot of, with respect there. But yeah, those are the samples from the last generation. It's going to probably look pretty similar. So we do have information.
And that's
collecting samples
wonderful,
in that area.
especially for Point Lay where you're near shore, because we really don't have any near shore monitoring and surveillance. That's what I'm getting at. And we'd love to have more of that in the region. Thank you so much. And thank you for letting as much science as humanly possible happening on your cruise, because you guys are the only ones getting it. Without it, we'd be zero. So thank you so much. That's a question really for these guys. Hope that helps.
We are hoping actually to start some biological sampling and harvest monitoring in the spring of 2025. We do have
Wonderful.
some seed funding that was recently awarded to that, and we're trying to apply for some other funding too. And that sampling will be done in consultation and direction with the subsistence communities, what their priorities are for sampling. And so we've made that commitment. So we're hoping the sampling does, get fresh samples for the abundance estimates at St. Lawrence Island, but also could address any other concerns and disease and pathogen surveillance from fresh harvested animals. That was the model we had for years. We've had 3 or 4 year hiatus. We do miss it as well. It's not a conscious choice. It's just a funding reality. But I think we'll be back.
Wonderful. There's some news. Thank you very much, Joel.
Yeah.
You answered that. So maybe we can start looking at local samples, local animals in 2025.
Yes.
Harvested near shore. Thank you. That's awesome. That's helpful. All right. With any other questions, we've given it to these guys. They're standing up for this whole thing. They probably want to sit down, but
Or eat a dinner.
or eat a dinner. Yeah, well, they came in off the plane. They chose their path. They chose their path. No, really, thank you very much. So for next week's Straight Science, we are having we're probably gonna have something later on. It was gonna be marine mammals co-management, kind of what is it and where are we with it? But that is gonna be rescheduled until probably in later June. The next Straight Science that we know we're having will be on the 19th of June, and that will be the US Coast Guard District 17 Command Chief, who will be laying out for us all that the Coast Guard will be doing in the region during summer 2024. And that will be on the 19th, unless otherwise you can just, there's stop share, if that's what you're trying to do. I don't go there, but so that'll be next week. And just so everyone knows, I'll come over here, just so everyone knows, this tonight, though it was a marathon, this tonight was the 250th Straight Science. So that's, and it's nice to have Bob Metcalf in the house because he, Carol Gales, if you're still on, Carol, and myself, we all started it, but it was really Bob who brought the idea to life, and we've been keeping it going. So thank you all online and in the audience.
And you're competing with Nome Belts High School graduation.
And we're competing with Nome Belts. What, that parade went by, and I was so glad it went by before we started, because it's all.
Joel wanted to go on the parade. He's like, we need to go to Straight Science.
Yeah. So anyway, thank you all. We'll see you next time. Bye-bye, everybody.
Thank you.
Thank you.